Okay, Hangout is live. All right, so I picked this. Actually, Andy sent me this paper or this chapter, and I thought it'd be a pretty good place to start since everything comes back to the glutaraldehyde and the chemistry. So that sort of is a good, definitely, especially for me, a place to start. Start at the basics and move on from there. And especially with the paper we're trying to write, because obviously it's all about the chemistry of glutaraldehyde. So this is from a electron microscopy textbook, and I know I had the reference. It's just a couple years old, right, Andy? Like 2008.
I think this is actually a quite old book.
Okay.
It's most of these books are pretty old, and they go through multiple publications. So the latest publication
Okay.
might have been 2008, but I think Robert was saying that this is in the 1980s somewhere.
That makes it because there's no references past like the late 70s, but that's okay. So it's a reprint. That makes sense. Okay. So I'm going to hit screen share here again, and let's see. Okay. So just some basic notes. So it's been about 20 years since I had biochemistry, so a lot of this I had to really review. So to start at the very beginning, what exactly is an aldehyde? And that basically is just what I'm showing here, just a molecule where you've got the carbon and double bond oxygen and hydrogen and then one other group. And can everybody see my little mouse pointer? So if I want to...
Yeah, I can see it.
Okay. So that's pretty good place to start. And the simplest aldehyde is formaldehyde, which comes up a lot, and that's just where the R group is just another hydrogen. So this is glutaraldehyde, and for people who forgot, when there's nothing, that's a carbon. When it's just there's no specific atom in there, that's carbon. And carbon, of course, is the 4 bonds, and so like this is a carbon, you got 2 bonds to oxygen and 1 to hydrogen, and this little model here of the element. And so when I first was learning about this stuff, I thought, oh well, this is easy. So by cross-linking, you mean you get one, I think these are carboxyl groups. So you get one protein on this side and one protein on this side, and that's the end of it. How very nice, but it's a lot more complicated than that. And so this paper kind of really got me into the chemistry. So just a couple of random points. So glutaraldehyde is actually used as disinfectant. So for a lot of people who want reversible cryotics, and we're trying to tell them this is a very deadly substance. I think you could even say, well, look, you use it to to kill microorganisms and viruses. So it's a very definitely not very toxic element. And so glutaraldehyde first came to, or I guess was used in tanning before anything else, and that makes sense because the whole point of tanning is to strengthen the leather, and so that probably has a lot to do with cross-linking the proteins, and that makes sense. And then it was 1963 where its usefulness for electron microscopy was discovered, and so this is something that I kind of just been learning too. So you first do the glutaraldehyde to link the proteins, but then the second step is the osmium fixation. And I think you typically use that for electron microscopy, but then you don't necessarily want to do that if you're looking at some of the more molecular biology kind of things. So is that right?
Yeah, that's absolutely right. I think later on in this, and Robert, you should probably take over here, but I mean, essentially, the osmium tetroxide is to stabilize the lipids, and the lipids are going to be extracted en masse and dehydration steps. And so if you're not going to a dehydration step, as we would not be going in aldehyde stabilized cryopreservation, then you don't need osmium tetroxide. Is that right, Robert?
That's right. The first fixatives that were used for electron microscopy was osmium tetroxide, and then later they added the glutaraldehyde as a first step. But really, the osmium's mostly only for electron microscopy. So if you're going to do histology or if you're going to do fluorescent imaging or anything, then you don't use it.
Okay, and that's, I guess, an advantage of the ASC is that you don't have that step. You preserve, just with glutaraldehyde and then the cryonic part.
That's right.
So that's a big advantage with that.
Although
Okay.
I like to think of it as postponing the decision to take that step.
Yes,
So
because
it might be in 100 years they want to take that step, but they would probably take it with something that's better than osmium tetroxide, or at the very least they would take osmium tetroxide in the most optimal fashion for preparing for final electron microscopy, if that's the way to go.
Yeah, and then 2 points to that is it's not like there's just one osmium step, right? There's, do you use ferrocyanide in osmium? Do you use uranyl acetate as well? After this book was published, they developed the Gram-Knot protocol that's really useful that uses uranium and uses reduced osmium. So, and then there's a million different types of resins that you can use too afterwards. So you can just defer all of those decisions till later.
Hey guys, 1 second. Hey Michael.
yeah.
Do you have mine selected right now? Like go to your Hangout and unselect mine. Yeah, you see how it says Kevin there? Select yours. The one to the far right. There you go. Because right now everybody online has my picture
Whoa.
up there.
Okay.
Recursive loop.
Okay, but let me go back to the okay, can everybody see the PDF again?
Yeah, so now that's full screen for everybody. Thanks, sorry about it.
Gotcha. Okay. So yeah, this is just warning, glutaraldehyde toxic, all that stuff. So a lot of interesting stuff about the chemistry, which other than Robert, I don't know that the rest of us need to know too much of that. Impurities and whatnot. Although it sounds like that's can be important. But a big thing that I want to talk about is the polymers, because it seems like that's a big issue. And if it turns yellow, then it's starting to get too many polymers. So that's something I saw.
Except one thing though, real fast.
Sure.
There's this 1979 paper by Robert Schultz where they just kind of cooked the glutaraldehyde for a while until it turned yellow, like dark yellow, and then compared it rigorously to perfect monomeric purified glutaraldehyde, and they found essentially no difference, but the small difference they did find suggested that the polymerized one actually worked better as a structural preservative than the monomeric one.
Okay, well, yeah, that's my next thing is I'm gonna actually go through the paper and I want to talk about some of these different possible structures, because it sounds like, I guess if this was written in the early 80s, at that point, I'm not good here, it sounds like there was a lot of controversy about what is most important. Is it the polymers that are really doing most of the cross-linking? Is it the individual molecules? And so to me, that seems like a critical question. So let's see if I have any other notes that I want to talk about now. Oh yeah, okay. So ask if the group agrees with this. This is from the paper. Although glutaraldehyde is used extensively as a cross-linking agent, not only in electron microscopy, but enzymology, et cetera, the nature of its reaction with proteins remains uncertain. Since the reaction products of glutaraldehyde with tissue proteins have neither been isolated nor identified, the chemistry of its reactions with proteins has been the subject of much debate. So is this still true today? Is that something that in the last 20 years people have resolved, or?
I'm also very interested in that question.
I haven't personally found a definitive, like, this is for sure how glutaraldehyde fixes proteins resource yet.
Okay, because that's what I want to that's the main part of the paper I'm going to talk about when I pull it up here, because I talk about at least two major controversies related to the cross-linking. Is the proportion, number one, the proportion of monomeric versus polymeric species in commercial glutaraldehyde solutions, and then more importantly, point number 2, the extent of cross-linking carried out by the 2 types of species. So let me jump to the paper. And I don't know, I mean, maybe this isn't important at all, but it seems to me, if you've got a huge chain of molecules linking proteins together versus, with the one small glutaraldehyde linking things together, that could make a big difference. So, with whether it's tertiary structure of the proteins or, some of that real small scale, stability. So I guess that's why I'm kind of interested in that.
So, as a quick question, because I was reading this as well, and very kind of fascinated that it says that multiple glutaraldehyde monomers essentially linking up together to make one cross bridge between one area of a protein in itself or 2 proteins. If that was if I don't see why that wouldn't occur in both the pure monomeric glutaraldehyde, if you pump it in without it having polymerized. Is it essentially that it takes longer for those polymers to form, and that's why it's better to start with some of those polymers than with the pure glutaraldehyde.
Okay, because you actually need a lot of those polymers to link the proteins.
Yeah, I'm kind of just like Robert. It goes out of its way to say that, hey, the polymerized version kind of works better, and I'm trying to get my head around why there would be a difference.
Okay.
What I've been thinking is that maybe, like, if there's more longer things, then there's more potential proteins that can be cross-linked, right? Like things that would normally be too far away for a single molecule.
Yeah, that's what I was thinking. Yeah.
But yes. I don't know.
Yeah, well, and the one, actually, let me jump back to there was one thing I wanted to. So one thing that was interesting was they did some studies of hemoglobin, and actually looking at how many glutaraldehyde, molecules are there, and let me see. On human blood cells, depending on the glutaraldehyde polymerization, between 100 and 300 sites in hemoglobin will react with the dialdehyde. Approximately 200 molecules of glutaraldehyde may be involved in cross-linking each molecule of hemoglobin. And the hemoglobin is just made up of what, maybe 300 amino acids? So that's like almost every single one. 2 out of 3 being fixated or fixed with the glutaraldehyde.
To be fair, like I've actually done experiments on this where I have glutaraldehyde that I measured the spectrogram of, right? And I saw like, oh, okay, this one is like 80% polymerized, right? Only 20% of the signal comes from it being pure.
Uh-huh.
And then I bought this super special, fancy, purified glutaraldehyde, right? And I measured it immediately before the experiment, and it was all pure, right?
Okay.
And I've done multiple trials of this, right, with different rabbits, and I couldn't even really tell a difference at all. This effect is like super subtle. So I mean, like some papers claim like, oh, it's a little bit better with the polymerized one, but it's very subtle. I mean, they both work fine. So
you're saying in the studies you did with the rabbits, you did it when you looked at it before and after, it was mostly the monomer. That was 90 whatever % of what was in there.
So you can buy purified glutaraldehyde.
Mm-hmm.
It's much more expensive, and so what I did was I compared a rabbit that I'd fixed with the pure monomeric glutaraldehyde versus another different rabbit that I'd fixed with glutaraldehyde that was just standard biological grade, and there wasn't any difference.
Okay, so that's the question of, does it naturally form the polymers that are needed on its own?
I don't know, but when it comes to ultrastructure, it's like there's basically no difference.
Okay. Well, so whether you start with the pure form or not.
Yeah.
Okay. So let's just get a little to the chemistry here. So here's just when there's a couple of different options. This is just one version of how glutaraldehyde forms a polymer. So the aldehyde reacts with the water, and you get a couple of hydroxyl groups, and then the two molecules of glutaraldehyde join and form like the ring structure, and then they can link up. The hydroxyls can sort of link up, and you can get multiple glutaraldehyde molecules, as many as you need in this kind of polymer. All right, and then there's a question of, well, is this a reversible reaction or not? And that could be important, and that I'll get to that too. So I don't necessarily want to go over. Let me just skip around each and every. I want to get to the protein part. So here's another potential polymer of glutaraldehyde, and so you've got the ring structures combined with just the straight molecule. I'm going to jump to, I think, page 19, where it gets talking about the proteins here. Okay, so here's the start where we're talking about, okay, so forms polymers. Okay, well, how does it, what's the chemistry of reacting with proteins? So here is where you learn about Schiff's bases, where I had actually checked that out in Wikipedia here, and it all that means is just a Schiff base is just something where you have what is it like here with the protein? You've got a carbon with the nitrogen, hydrogen, another group, or something like that. It just means a certain number of things binding with the carbon. So it wasn't anything more than that. So here's one theory. Okay, so you've got one molecule of glutaraldehyde reacting with a protein, and it always reacts with the amine group, the nitrogen group, part of the proteins, and so it links it together, and presumably you could link it in both sides. But I guess Mullen et al. Are saying this might not be really what's going on, because this is a reversible reaction. So it's not going to be anything that necessarily would be stable. So it has to be maybe something more complicated than just a protein binding to each side, because here in step 2, well, let's see. So step 1, you get the protein binding, but then you just go backwards because it's a reversible reaction, and so it would not necessarily be something that's stable. Does that make sense? I explain that.
I think it makes sense. I have a question that you should probably wait until the end to answer, but I'm dying to understand what the implications are of reversibility, and essentially what the heck is going on if you were to leave such a brain in glutaraldehyde in the liquid phase at, let's say, 4 degrees Celsius. But don't derail the conversation now. I just put that in your to-do list.
Okay. Well, yeah, that's a very interesting question about would just slowly reverse itself, and I think that the answers are going to be no, because of some of the other proposals about what's going on that forms a non-reversible reaction. So okay, so let's pan down to the next one. And so here's where it gets to a bunch of debates. There's a lot of different reactions here, where somebody says, oh, it's this way, and someone says, well, no, it's, here's what's really forming the bonds. So, okay, so here we are. So Peter and Richards are saying that, okay, the most reactive species is, and here you get the polymer structure. Okay, and they're saying it's proteins reacting to not the carboxyl group on the end of the molecule. They're saying no, it's reacting to the middle molecule here, and what do they call it? It's not the terminal aldehyde, I guess. Okay, that's what so this is like the terminal aldehyde. It's not that, because that's a reversible reaction. So what it is the protein is binding to a carboxyl group in the middle of this long polymer that's not on the end, and somehow maybe that's a more stable bond. So that's what they're suggesting.
Let me ask a really stupid question. In general, what is the difference between a reversible reaction and a non-reversible reaction? I assume it's energy, uh, but is that
Yeah, I think and I think here they're talking about like reversible with like like the reaction is going to go both ways at the energy level that you're doing the mixing or whatever. So because I guess every chemical reaction could be reversible if you pumped enough energy and whatever into it. So yeah, they're talking about reversible versus non-reversible at the same the same test tube, whatever room temperature, whatever you're mixing this stuff together. So that's how I think they're using like reversible and irreversible reaction. Asking whether does that matter? I mean, would the reverse reaction happen in the same test tube, with the same energy level?
okay.
But I mean, I'm throwing this out. I'm a long, long way from being a chemist, so I'm just that's kind of what I was reading to the paper. I could be totally off there, but so yeah,
You've
so
got
that.
Andy on the line too, right?
Okay, yeah, yep. Andy's on the line, so he's much closer to the stuff than I am. So that was my understanding of that's what this is.
I don't think I'm much closer actually, but my take on it,
Okay.
at least the use in this paper, is that reversible means like empirically able to be reversed with treatment with acid or urea or something else like that.
Okay,
so
yeah, I did.
Okay, yeah, I did. Like whether it's empirically able to be reversed.
Okay, yeah, I do remember reading about that with the with the acids are different depending on the pH level.
So theoretically, they could say it's irreversible because they haven't found a way to reverse it yet. I mean, if that interpretation is correct, we should probably get a chemist here.
Okay, well that's interesting. Yeah, because I definitely remember them talking about the, being reversible if you change, add acid, change the pH. So, okay, so all right, so here is.
Just one other note. I think it's an aldehyde group, not a carboxyl group.
Oh, you're right. It is an aldehyde group. You're right. Okay, so all right, so that's proposal number 2 up there. So let's keep going. All right, so Richard and Knowles, 1968, aldol condensation. Okay, then then you get into more and more of the chemistry here. So they're saying, okay, they're these 3 different glutaraldehyde molecules join up and you form this double ring structure. And I'm going to keep going down here because, okay, this I don't understand these 2 at all. I tried to follow the chemistry here, but let's skip to the, because they'll come back to that. So here is, okay, another proposal where, and this one's a little more straightforward. Okay, so you start with the glutaraldehyde, you form the ring structure, and then these ring structures join, and then it's the R group here could be the proteins to each end, and so they sort of link in this chain. That could be, and then assuming again that this is a less reversible reaction, more stable reaction, and then I'm going to keep panning down here for. Okay, and then this. Then you get into even more complicated, and I assume these are all just polymers of the glutaraldehyde, and oops. Okay, and then you form these even more complicated structures where you get the this triple molecule with the 2 rings and the proteins attached to each side. So, and if it was, and I don't know if this can keep going or if you're just limited to one of these structures linking 2 proteins, or can you, add another one of these together? And they didn't really get into huge detail about this reaction.
Well, they say something about on average each binding point, I think they called it, has on average 3 monomers? Is that constraining how many of these could be involved?
No, say that again. So each.
Yeah. So somewhere in the paper they talked about essentially 3 glutaraldehyde monomers on average are used for each binding point. I have no idea how they actually calculate that.
So for like each amine group and like an amino acid, like each potential binding point in like a protein.
Yeah, I don't know. I'll
Okay.
see if I can find it.
Well, that's not too bad because I mean that hemoglobin one said there was 300 molecules for hemoglobin, which has about 300 amino acids. So, well, I guess that's more like a 1-to-1, but that's not too far off. I mean, that's all right. Well, let's see if there's anything else. Okay. Well, then they kind of throw out even more fancy structures where I start to kind of lose it. I think this is a. Let me go down to the bottom one. Well, okay. So then, throwing out some of these at the end without too much explanation, which kind of confused me a little bit. I think that's the end of the. Yeah. Okay. So that's the end of the protein section. So I guess maybe the take-home message for me at least was, wow, there's a lot of complicated chemistry in here, and is there any research out there that maybe clarifies this, and maybe even importantly, does it really matter? I mean, if we know empirically that you do the rabbit brain and you look at the structure, and it's the connectome is there, then does that all really matter, or does it matter more for, well, what if we wanted to measure the DNA, or what if we wanted to measure each neurotransmitter in the vesicle, or the tertiary structure proteins? Is it only important for that, or does it even matter for that? And I think that's kind of what we're trying to find out with the review paper that Andy and Naveed and a bunch of us are trying to go through from the molecular level all the way up to the entire tissue level of what's what's really going on, or what how well is this preserved, I guess, with glutaraldehyde. So for me, that was it. Your take-home message: wow, there's a lot of complicated chemistry here that honestly, I'm not sure how important it is.
Well, the number one question, if I can suggest, the number one question that has to be answered by this paper, or in general, is there information loss, and where is that information loss coming from?
Yeah.
So is there?
Information loss all the way down to the molecular level, like tertiary structure proteins or DNA, or really.
And I should also say, okay, we have to. Okay, so there's information loss, and then I think that, to be honest, we need to have another category, because there's the possibility that the information is still there, but it is so hidden that it really makes certain techniques unable to see it. So, for instance, if we are assuming that that there is information that is necessary to get in the proteins, what proteins are where, and that the only way that you can get to that information is by the standard technique that we use today, which is immunocytochemistry, then different levels of glutaraldehyde are obscuring that to different degrees.
Yeah. You say different levels of glutaraldehyde. You mean the?
So for instance, a lot of the so one of the key questions that I had in reading this was some of the stuff that they're saying about conformational changes or the enzymatic activity that's preserved or the ability to use immunocytochemistry, a lot of that is dependent upon the concentration that is used.
Okay. And do they know how, like, less is better, or I mean, is that like less concentration?
Okay, so there's a couple things at play here. So earlier on, they talk about how
The
if you
discussion,
don't use that much
Robert.
glutaraldehyde. Oh, is there a problem?
I can hear you. I don't know. Let me.
So like early on, they were talking like if you don't use too much glutaraldehyde, then you can even get chloroplasts to just still work without any trouble. And that's much less glutaraldehyde than what aldehyde stabilized cryopreservation would use. And then on the high end, you use a lot of glutaraldehyde, it can cause difficulties with subsequent immunolabeling, not because it's destroyed the antigenicity targets, but because it creates a bunch of false targets everywhere because there's so many free aldehyde ends sticking out everywhere. Now, so for a long time, people used formaldehyde or they used a small amount of glutaraldehyde if they're going to do immunocytochemistry to avoid the background staining. But that doesn't mean those proteins are actually gone. It just means they're masked.
Yeah.
And with the advent of the borohydride methods and the switch protocol, that's no longer the case. In fact, if you look at the switch protocol, right, where they're using pH to control whether that glutaraldehyde is going to bind your immunoprobes, now you can actually get more antigen targets with glutaraldehyde than with formaldehyde. So more, I think, is probably better, although it is going to totally shut down a lot of enzymatic activity that might have been there before.
So in the switch protocol, so they'll use the glutaraldehyde solution and then change the pH to sort of control how much is cross-linking. Is that interesting? Okay, now would that work for? Well, I mean, I guess so. You do the whole brain. You do the glutaraldehyde and you freeze it. Could you change the pH enough so that you could do different, immunochemistry kind of things, and then change the pH again, and then do the osmium, and then do the, the scanning?
Oh yeah, like if you want to do some correlated microscopy stuff. Let's see here. Yeah, why not? I mean, if you had a slice of brain tissue, right, you should certainly be able to put in some immunolabels, and then use a confocal microscope or something to get some data off of that, and then take that slice and then embed it for electron microscopy. Sure, you could do that.
Okay, well that would, I was gonna say that wouldn't. I guess the dream would be to totally reverse it, but that you can't do that with the pH. You're just doing a little bit of leeway with adjusting the pH, but not reversing the glutaraldehyde and then thawing out the brain or something,
No,
right?
yeah, I'm just saying the reason people say, oh, it's glutaraldehyde gives you a lot of trouble for doing certain types of immunostaining is not because at a higher concentration of glutaraldehyde you're destroying anything. It's just that at a high enough concentration of glutaraldehyde you can't use the immunolabels anymore because they get bound up with the glutaraldehyde.
They're sticking to the glutaraldehyde. Oh, okay,
Yeah.
makes sense. That,
Can I interrupt real quick?
Yeah.
Hey guys, I'm sorry I have to leave right now, but I've been taking notes on some of the questions that you guys want answered. For example, a specific one was, is there any denaturing of tertiary structure by GA at 4 degrees at room temperature and at ASC temperatures? And so if anyone else wants to record additional questions, I've been collecting people on LinkedIn while we're having a call, while this has been going on, using searches like glutaraldehyde chemistry. I found a really cool guy who's a protein polymer chemist in San Diego, and I'm happy to send our questions to some of those people and see if any of them might be interested in helping us answer them. So I think that's a little piece that I could play if someone else is willing to, record the very specific questions that you would like to be sent off to some of these potential experts that we could bring into our group.
Well, yeah, and I definitely think it's. I mean, somebody talked about a Google Doc, like summarizing, maybe the meeting or or the high points or the unanswered questions. So yeah, I definitely think that was good. I mean, I guess this was kind of a test run, so just to get the thing going, and it seems like it worked okay if I just somehow had thought to reboot my computer. I mean, it seems like it's a pretty straightforward, it's really not that hard, but one time you want it to work, of course, it doesn't work. But
that's great. Yeah.
And then Kevin, are you still on?
Yep, sure am.
So you're saying we are limited to. Well, I just want. I mean, well, obviously we'll get back to the science.
9 participants, but unlimited viewers.
Okay, well, that's probably going to be good enough for most of the meetings. But what if there's like a subscription that lets you do more or something?
Yeah, there's a pro account that lets you go up to, I think, 14 or 16. I'll have to check on that. It's the business account.
Okay, but everybody can watch on YouTube and everybody can type in comments.
Yep.
Well, that's probably good enough.
I think we can cross that bridge when we come to it. If we come to it, I think this is great. I can't wait to have more of these. I have a ton of questions, and I think that we've already identified several other papers that we probably need to devote some time to and make sure that we're all reading, like the switch paper. There's some other obvious questions that we haven't talked about, and which this chapter didn't talk about, which is the not the chemical effects per se of glutaraldehyde, but the structural changes that occur in the brain, which I would argue are probably more detrimental to the information, loss of extracellular space. Getting a paper like the Cragg paper and really digging into and a recent Graham Knott paper that was devoted specifically to extracellular space and how that changes with fixation. These are all things that we should know.
Yeah, no, I agree, and I definitely want to learn a little more about that switch procedure. That sounds really interesting.
So would it be helpful if I want to just you started a little bit late, so I feel that we can run a little bit over time.
Yeah. I'm good.
Okay, so I think if we're concentrating on the ASC paper, which I have to say, let me just back up for a second. Okay, so my take on this is that if glutaraldehyde hadn't been discovered in 1963 or whenever, its effect on as a good fixative for electron microscopy, if it hadn't been discovered, this would be essentially like a magic information preserving chemical, right? Yeah, I mean we have this thing in the back of our minds that says, oh, you put in glutaraldehyde, you get a corpse. And what this paper is really, what this article, this chapter of a book is really, the take-home message from my point of view was that you shouldn't be thinking of this as destroying the chemical nature of the brain. You should be thinking of it as lightly locking down the chemical nature of the brain, so that the things that could be enzymatically, metabolically destroyed can't get to the things that would do that destruction. So they're all there. They even have their enzymatic activity still, but nothing can get to anything else. And now if you borohydride the system to get rid of any loose bonds and then flush in your antibodies, lo and behold, you can still peruse this structure for its chemical nature. And so essentially what this adds up to two statements. Glutaraldehyde lightly locks down everything. If you just left it in that form, it's lightly locking down stuff is not enough. You wouldn't want to be storing a brain like that for decades because it's just not, there's still so much room for chemical reactivity and probably reversibility. And so ASC comes in, stops it in time, and you can then drag it back to exactly the point where it was lightly locked down 100 years, 200 years in the future. And so I think that that's kind of the take-home message that we want to focus on, and we want to focus on stuff like, okay, for ASC to work, you have to get to this concentration of glutaraldehyde for this amount of time, and that out the research that says now 3% is consistent with this level of protein structure preservation and this level of immunoreactivity identification. So it's that 3% is consistent with that. And the other aspect is, okay, glutaraldehyde has all these other structural changes, and it doesn't tie down certain molecules that are washed away during the ASC procedure, not during dehydration and stuff like that, but during the ASC procedure, and really nailing in, because there's all these papers that talk about the chemistry of glutaraldehyde and how well they do on plant tissue and stuff like that, and that's, to a certain extent, not relevant to what we're discussing. We're trying to say, look, if you had this magic ability, which Robert has developed, to stop a glutaraldehyde fixed brain 4 hours after you started perfusing it, what level of information have you preserved, and how easy is it to access that information?
Yeah, I mean that's absolutely, that's all the information that we want to know. I mean,
so does anybody have any other questions?
or Andy, any thoughts, since you kind of pointed me to this chapter?
Yeah, that was interesting. Thanks. Awesome. I mean, I agree with what everyone said. It's all good stuff. Ken had a good summary. So.
Yeah, and I think, you know, the paper that a bunch of us are trying to get together is you know, trying to see what's out there, you know, like a literature review, just try to you know, answer some of these questions the best we can. And I actually, since I'm away from the chemistry, I'm doing the tissue and organ section, and I've been, you know, reading some interesting stuff about, you know, they actually use glutaraldehyde in some of these. No, it's not like an organ, but, you know, like for a structural basis to you know, do some kind of like bioengineering. So in terms of, you know, keeping the tissues together, I mean, it even increases the tensile strength and stuff. So, you know, at that level, I mean, I think that
and they actually use glutaraldehyde treatment. This is a somewhat of a question, but I believe they use glutaraldehyde treatment for parts of pig hearts.
Yeah, the valves or something. Yeah.
They do, but it's a very low concentration of glutaraldehyde.
Hmm. So to get the strength part, you just need a low level to actually get the collagen together.
Something like that.
Okay, that's interesting. Yeah, because I did a little paper. I found some on the pericardium of the pig, and I think in a cornea. There's a couple of tissues where they're actually using it, and definitely with collagen to build like a scaffold or something for some of these things they're doing. But I think that's the easier question than, okay, but what about the tertiary structure? And I'm leaving that to Andy to figure that section out.
So I think the other question that is possibly left unanswered, Robert, maybe I missed this in the paper. The real answer to why glutaraldehyde pre-treatment allows the cryoprotectant not to dehydrate the cells. Have you had any further thoughts on that, or want to summarize kind of your thoughts on it that you gave in the paper?
Sure. It's just it's not really the glutaraldehyde that stops it. It's the fact that the blood-brain barrier has been permeabilized. So even if the cells were not actually fixed, if it was just cells, then it would not shrink upon the introduction of the ethylene glycol, because it's enough time that that 4 hours is enough to reach enough equilibrium that it won't suck the water out. It's just with the brain in particular, as long as that blood-brain barrier is in place, it's just not going to work.
But now I thought I remember reading in your paper, or maybe it was a discussion outside the paper, but I'm going to assume that was in the paper, that just the sodium dodecyl sulfate treatment did not, by itself, which would have opened up the blood-brain barrier, did not by itself eliminate the dehydration in the cryoprotectant?
No, that does work. If you put the sodium dodecyl sulfate, then it does eliminate shrinking whether or not you use glutaraldehyde.
Oh, really?
Yeah,
Okay.
in fact, the original experiments they did, at least that Yuri Pichugin did at the Cryonics Institute, were exactly that. Just the dodecyl sulfate and then normal cryoprotectant loading protocols, and he found that he could avoid the shrinkage with, of course, damage to the brain, but that was
exactly. That's what I'm asking. So, and feel free not to answer if you don't want to, because I know we're on live. But if you were not to put in the aldehyde, because it really comes down to, gee, what is the aldehyde doing? What are the good things? What are the bad things? What is the necessary concentration? Anybody reading a paper like this, or anybody reading your aldehyde stabilized cryopreservation paper from the point of view of saying, gee, we want kind of an optimal protocol for both structure and molecular information, is going to be asking, gee, what's the kind of minimum amount of stuff that glutaraldehyde is doing that has to be done or else?
Certainly, if you don't, so if you just use formaldehyde, right? Not even just like no aldehydes, but just formaldehyde, right? It's not good enough. The ethylene glycol just ruins everything, causes a lot of damage, and doesn't even get everywhere that it needs to go. If you don't use anything at all, right? If you just use like the sodium dodecyl sulfate and then go in with some cryoprotectants, if it's ethylene glycol, it's going to really do a tremendous amount of damage to the structures.
Okay, so you keep saying that if it's ethylene glycol, I need to ask, would this be different for the VM, whatever?
Let's say it's VM3, right? Like, it's still going to cause a lot of damage, but less. So SDS plus like VM3 would probably be your best shot, but it doesn't look good. In fact, Jordan Sparks has some things on his website where he did exactly that experiment, and the electron micrographs are not the best structure. So you need the glutaraldehyde to keep everything together.
Okay, so your interpretation is that is a very general one. That's what I'm hearing. This is a question, though. is that the fact that the aldehyde has stuck proteins together in the cytoskeleton, in everything else, that has made the tissue more robust to the few hours of perfusing in cryoprotective agents.
That's right. I mean, it allows you to do things like do it at room temperature even, right? So you don't have to do it super cold. So it buys you a tremendous amount of stability. But it does not prevent the shrinkage. In fact, originally I just thought I would do glutaraldehyde for do a cryoprotection ramp over a very long time, and that would be good enough to prevent shrinkage. But even 96 hours isn't enough time to do it, as long as that blood-brain barrier is intact. But just a little bit of STS in 4 hours is enough. So yeah, and actually, I take that back. I recently did some stuff with Boyden's lab, and I found that 2 hours is good enough.
Okay.
That's cool.
So you're saying the glutaraldehyde without taking down the blood-brain barrier will not prevent the shrinkage? Is that
That's right. Upon the
So
introduction.
wait, what do you use to destroy the blood-brain barrier? What was that you said?
STS, the sodium dodecyl sulfate.
Oh, okay. Okay. And then that lets the glutaraldehyde get in there, and then you're safe to.
Well, the glutaraldehyde's already in there. It allows the ethylene glycol to get in there.
Okay. And if that doesn't get in there, then you're going to get the shrinkage when you.
Yeah.
Okay.
Well, and I wanted to ask you one other thing. This is a little off topic, but Robert, they were talking about the calcium chloride being used to prevent myelin figure artifacts. And I looked up your paper, and if I'm reading it correctly, you used the calcium chloride in the pre-wash, but not in the actual fixative.
I've tried so many things. I've tried calcium chloride in the fixative. I've tried magnesium chloride. I've tried copper compounds. I tried acrolein. I tried just a bunch of random stuff, and nothing stops the myelin from unraveling. I've only found one thing that stops the myelin from unraveling, and that's permagnate with no aldehydes whatsoever, and it just destroys everything else. But what's really interesting is if you do permagnate fixation and then you follow it up later with aldehyde fixation, it unravels the myelin. So, like, as far as I can tell, it really is the fact that aldehydes themselves intrinsically damage myelin in this way. And if you touch them with aldehydes, then you get this effect.
And they had a very brief discussion of that in this chapter. Did they give a reason for that? I can look it up.
I've never seen a good reason for it. Like, I don't really understand it either. I will say the myelin structure is more complicated than people think it is. It's not just stuff wrapped around. It's like you got all these incisors going on, and it's like there's actual channels that can expand in myelin, and then it will look like unraveling. And so my best guess is that the process of the glial cells swelling is also causing these incisors, which are connected to the glial cells, to swell, and then it looks like the myelin's becoming unraveled. But
Robert, I mean, I'm talking about myelin figures, not myelin itself. So the whorls that are inside some of the cells in your.
You're talking about those things. Okay.
Yeah, those are myelin figures, correct?
Yeah. I thought you meant the myelin unraveling. Okay.
That's what they talked about with the calcium chloride and etc.
You know, I've never managed to totally suppress that. I haven't seen too much of an effect either way.
So what I was asking, I think you answered it, was you did try calcium chloride in the fixative itself, and that didn't.
Yes.
Yeah. Okay.
It doesn't do much. There's very little difference. I mean, even changing out the buffer systems entirely to other random buffer systems, it doesn't do much. So yeah.
So I mean, should we be okay? So as a I'm not a skeptic. I can't claim that anymore on this. But a skeptic would say, what the heck is going on, making all of those weird clusters of lipids. And essentially, I guess what this is saying is, look, if you're using glutaraldehyde, you're not doing anything to lock down a certain class of lipids. You know, there's some phospholipids with amine groups that they talk about, and maybe those are locked down, but there's just a whole bunch of lipids that are out there that are hanging loose, and they can do some weird stuff. And another question would be, would that be ongoing if it sat there for a while, or is this basically happening in the first 30 seconds of the cell dying?
Well, that being said, aren't there myelin figures in freeze substituted brain tissue that wasn't fixed with aldehydes at all, that's just instantaneously frozen? Not as many, but they do exist, right?
Are there?
I believe, I'd have to look at the papers again, but I believe I've read about that, and that they do exist. There's just less of them.
Okay. Well, then that's.
I'm not convinced they're entirely unnatural structures. I don't know what they're supposed to be for, but yeah. Like I believe I've seen them in. I'll have to look at that online, but I'm pretty sure I've seen them in freeze substituted brain tissue.
Well, in any case, I think that this is. So I'm looking at this from a what would a reviewer, what would a somewhat open-minded neuroscientist, biochemist, you name it, medical doctor, what kind of questions would they ask about the ASC procedure that has not come through in the existing paper? And I really think that should drive what literature we're tackling, and I think it is. Well, cool. I think maybe we should call it. Does that sound good? Thank you very much, Mike. This was awesome. I learned a lot, and like every other journal club that I've ever gone to, and it's mandatory.
Yeah.
I learned a lot during the discussion, but I probably learned more because of the reading. So just the fact that it's not even just the people that were able to join us tonight, but the people that read the article that you selected. I think we should keep this going because it's going to educate us a lot.
Yeah, no, I'm the same. I feel the same way about journal clubs. I mean, they're really, I always learn a lot, and yeah, so I think Tuesday works for me, so let's keep it going. There's a ton of papers out there, so. Yeah.
So I'm gonna sign up at some point. I think we should all beg Robert to sign up to give his own paper.
Oh yeah.
Yes.
And then
you'd be willing to do that, Robert?
I would, although I want to put together a couple things for it, so maybe not next week, but maybe one after that.
Anytime.
So we're doing it monthly, right?
What's that?
Oh, next week is the regular meeting, I think.
Yeah, but we're doing this monthly, correct?
Yeah. Next week. Oh, are we doing this monthly? Well, I thought we were talking about doing it.
No, well, so the idea was to do this every week that there is not a BPF meeting.
Okay.
And at the same time, I assume that very few people will join. That's not the point. The point is to when somebody says they're going to do a paper, the people that are most interested in that paper will read it and join in. So Kevin, if there's a way to make that happen in such a way that it is not required for you to be holding our hands, that would be the best way to make that happen.
Yeah, no, it's just training. If I jump on with the people beforehand and just train them how to do it, but I don't mind sitting on as well. It's kind of an honor and a privilege learning from you guys. I mean, some of it, I mean, it's absolutely a different language. I was following to the polymers, then I'm like, okay, I need more information here. So I'll get caught up over the next couple that we do on these. So I appreciate the opportunity, and I'll be on for most of them.
Well, yeah, you saved me tonight, Kevin. I would not have rebooted it, so it would have never worked. So is there a way? Well, we'll talk about the details, but it seems like I messed things up by I thought I pre-invited everybody so that they just click a button and get on the Hangout, but it seems like that's a little more complicated than that. So that's
you invite us to the event, but we can't actually participate in the event. So
I gotta head out, but thanks for the meeting. Talk to you guys soon.
Yeah.
okay. See you later, Andy.
Thank you, Andy.
Okay. So I invited you to the event, but then I got to invite you. Yeah. Okay. Well. All right. Well,
I guess the event's
that.
just for listening.
Hey, Michael, you can go ahead and hit offline.
Oh, okay.
And just say bye to everybody that's live right now.